Hi everyone. So thank you for that introduction, Liz. I'm a senior postdoc at WEHI, and today I thought I would tell you a bit about the project that I'll be working on for my Cure Cancer Fellowship, and just starting with a little bit of background. Okay. So in Australia, we have about 20,000 people diagnosed with blood cancer each year, and this makes up about 12% of all cancer cases. And blood cancers are not only quite common, but they're also the third leading cause of cancer death. And so my research has really focused on blood cancer and improving outcomes for patients. So even though we've had a lot of really exciting advancements in like targeted therapies and immunotherapies, there are still many subtypes of aggressive blood cancer which have really poor prognosis. And for some of these patients, we really don't have any effective treatment options. So some examples are acute myeloid leukemia, where you can see the 5-year survival rate really hasn't improved in the last, almost 2 decades, as well as here an example of a quite rare but also quite deadly lymphoma. And so, as well as having particular subtypes of blood cancer, which are very difficult to treat, we also know that there are particular mutations which can make blood cancer difficult to treat. And one of those most common is mutations in the tumor suppressor protein p53. So it's clear that new treatments are required, and a lot of my research has focused on targeting apoptosis as a therapy for blood cancer. So just to briefly introduce you to this pathway, at the bottom of the pathway, we have the pro-apoptotic effector proteins, BAC and BACs, and these oligomerize to cause holes in the mitochondrial outer membrane, which leads to the release, activation of caspases and apoptosis or cell death. And in our cells, these pro-apoptotic effectors are usually restrained by pro-survival proteins. And then when we actually want a cell to die, we'll get sort of a stress signal that will lead to induction of these pro-apoptotic BH3-only proteins, which sort of inhibit the pro-survival proteins, freeing up BACs and BAC to trigger apoptosis. So this can be a bit complicated, but the easiest way to think of it is kind of like a scale, where if we have more pro-survival proteins, the cell's going to survive, and if we have more pro-apoptotic, the cell will die. And it's this balance between them that determines the fate of a cell. And so many cancer therapies that we're already familiar with, for example, chemotherapies, use this pathway to help to kill cancer cells, but many of them go through p53, which I mentioned earlier, which, for example, chemotherapy causes DNA damage, which causes activation of p53, which leads to induction of pro-apoptotic proteins to trigger apoptosis. But of course, many blood cancers have mutations in p53, rendering these therapies less effective. And so the goal of my research for the last few years was to try to figure out a way to get around this. So how can we kill these blood cancers without relying on p53? And so what we recently discovered is that activating a protein called STING can actually directly induce apoptosis in blood cancer cells. And so STING is a protein that's known for its role in sort of activating our immune system canonically, but we found this sort of new role where in blood cancer cells, it will actually kill them directly. And it does this by inducing expression of the BH3-only pro-apoptotic proteins. And what's really exciting and important about this is that it's independent of p53. So it doesn't matter if there are p53 mutations, we can basically go around p53 to initiate this pathway. And what's also really great is that we can combine drugs which activate STING, which already exist and are already in clinical trials for immune modulation, but not for this particular way of killing cancers. We can combine them with BH3 mimetic drugs. And this is a class of drug that has already been approved for therapy of some blood cancers. But unfortunately, despite being improved, many of those patients will eventually relapse. So we can use it as a way to improve this therapy. And the way that this works is that when we treat with BH3 mimetic drugs, we deplete these pro-survival proteins, which you can see here. And then we basically can boost this apoptotic signal and kind of tip this balance in favor of cell death. And so for the last few years, I've been investigating different cancer models to show how well this works, basically. So just a couple of experiments that I wanted to show you. This is in p53 mutant acute myeloid leukemia, which is a very aggressive type of blood cancer. And patients with this type of cancer, there's really just no effective treatments for them at all. And so collaborating with a group here at WEHI, run by clinician scientist Andrew Way. They've generated patient-derived xenograft models. So they take this AML from a patient and they put it into the mice and they can treat it and see whether, sort of as a preclinical test, sort of what we consider the gold standard for testing these therapies. And so this patient sample actually came from a patient who had relapsed after being treated with the BH mimetic drug venetoclax. So they're already resistant to venetoclax, which is one of the standard agents used to treat this type of cancer. And that's shown here in green, that there's no survival benefit with venetoclax. But when we treat with this STING activating drug, Diabzi, you can see that we get this really impressive increase in survival for this very aggressive type of cancer. And so even though this patient was resistant to venetoclax, and there doesn't look like there's much of a survival advantage when you combine the STING agonist with the BH mimetics, if we look in the bone marrow during treatment, you can actually see there's about 100-fold reduction in the number of leukemia cells in the bone marrow with that combination treatment. And so it looks like this could actually be a very effective treatment for this type of blood cancer. And then the other type of cancer that I want to show you here is the natural killer T-cell lymphoma, or NKT lymphoma. This is usually diagnosed quite late. It's quite a rare cancer, but for patients who don't respond to chemotherapy or relapse, there's really nothing else we can give them. And so here I'm showing you some pictures of cells cultured in vitro. And when we treat with DMSO, the cells are growing very happily. When we treat with the BH mimetic, you can see that some of the cells are starting to die in the middle of these clumps here, but overall still growing out. The STING agonist we see very similar, but when we combine both together, we get this really impressive apoptosis of those cancer cells. And I've spared you the gory mouse pictures, but I just want to show you some results when we put these lymphoma cells into mice, and then we treat them with just a single round of treatment, so just 1 week of treatment. I just want to note that usually when patients are treated with BH mimetics, this is an ongoing therapy. They will usually be on it for years. But this is just a single round of treatment, and we already see this huge reduction in the tumour weight, about half. And it also reduced the spread of this cancer to other lymph nodes in the body. And so based on these results, we've had a lot of interest from clinicians and also from biotech companies, and there's now a clinical trial in the planning stages for patients with acute myeloid leukemia, which will happen here in Melbourne, hopefully very soon. And so that's where my Cure Cancer Fellowship comes in. And the aim of this was to really support the clinical deployment of STING agonists for blood cancer therapy as like a novel kind of therapy, by first identifying other types of blood cancers that might also respond to these drugs, because we've only looked at a very few so far. And secondly, looking for biomarkers of response to STING agonists that we could use to help select patients for the clinical trials who are more likely to respond to the drugs. And this is really an area, a great unknown area, because at the moment we don't really know a lot about using this drug this way, because it is a brand new way to use these drugs. So one thing that we found in the course of performing these experiments is that the levels of STING protein in cells is actually already quite a good biomarker of whether cells will respond or how much they will respond to the drug. And just showing you some results here from some mouse lymphoma cells. It doesn't matter whether the cells have been CRISPRed to knock out P53 or not, whether they're mutant or wild type. What really matters is the level of STING, and this correlates very closely with the IC50 doses required to kill these cells. And so we know that STING expression is really important, and that gives us a really good starting spot to pick what types of blood cancer we think we should look at next. So I've showed you already that this drug works really well to kill acute myeloid leukemia cells, and you can see up here that the STING expression levels are really quite high compared to non-Hodgkin lymphomas. And this is at the mRNA level from cancer cell lines. If we break down the non-Hodgkin lymphomas, we can see that the B-cell lineage lymphomas generally have quite low expression of STING, but multiple myelomas and T-cell lineage lymphomas have higher expression. And so we thought these would be quite promising places to start. So a little bit about these other types of cancer. So these are also new research areas for me, and I'm quite excited to do something a bit different since I've focused mostly on B-cell lymphomas for my research so far. So multiple myeloma is actually derived from plasma cells, which grow normally in the bone marrow. And you can see here that when they overproliferate, they also produce these abnormal antibodies that can cause all the kinds of symptoms of this disease, which can include, bone breakages, bone pain, kidney damage, etc. And the current standard of care for this type of blood cancer is actually usually 3 drugs all at once, so proteasome inhibitors, steroids, and immunomodulators. And many patients will also receive a stem cell transplant if possible. Unfortunately, even with these treatments, the median progression-free survival remains at about 41 months. So it looks like there are new therapies are required, and we're hoping that STING agonists might be able to be included as one of these therapies. And then the other type of cancer we're interested in looking at is T-cell lymphoma, which is derived from T cells, either in our lymphatic organs or they can also be peripheral. There are, again, multiple different types of therapy for this, and there are also multiple different subtypes of T-cell lymphoma. And unfortunately, for the very aggressive types, most patients will not make it to 2 years. And when we're talking about p53 mutant lymphomas, T-cell lymphomas, I was actually quite surprised to see that many of these patients have, very, very low 2-year survival rate. So again, hopefully this is somewhere we could make improvements. So the plan for this aim of the project is to use a panel of human cell lines of multiple myeloma and T-cell lymphoma, and we want to look at STING protein levels and also response to STING agonists as a monotherapy, and then combine this with standard of care therapies in vitro as well, and then we'll take the most promising combinations to xenograft model testing. And we're hoping that this will inform the future clinical direction of STING agonists so that we know which other patients may be able to be included in these clinical trials. And hopefully in the end, we'll have a new treatment option for some of these most aggressive blood cancers. And so the second aim is to identify biomarkers of response to STING agonist therapy. And for this aim, I'll be doing a lot of whole genome CRISPR screening, which is something that I have a lot of experience with. So I'm going to do both traditional CRISPR knockout screens, where we find factors that confer drug resistance when lost, as well as a newer type of screen called an activation screen, where we can find factors that cause drug resistance when overexpressed. And to enable this aim, my lab has recently developed a CRISPR activation mouse, which has all the components needed for CRISPR activation. So we can take any tissue from the mouse, we can cross it with blood cancer models, etc., and then we can basically just put in our guide RNA to activate whichever gene we want. And this is something readily available, and we're also very happy to share this mouse with anyone who's also interested. And recently, we've also developed some new CRISPR activation guide RNA libraries to go with this mouse as well, which will hopefully be published very soon and also then available for everyone to use. Okay, so how do the screens work? Well, we do these screens in mouse lymphoma cell lines because we have a lot of experience with this system. And we take a library of guide RNAs and we transduce it into cell lines expressing Cas9. In this case, we use a well-known available library called the USA library, which targets almost every gene in the genome. We then split these cells up and we do multiple replicates, but for each replicate, we have a DMSO treatment, which is the vehicle control, and then we treat the other one with a STING agonist. And then we basically do multiple rounds of treatment until we outgrow the resistant cells. And so from the control cells and the resistant cells, we extract the DNA, we perform indexing PCRs, and then we perform sequencing to look at which guide RNAs have been enriched in the resistant cells, and therefore which guide RNAs are conferring resistance to the STING agonist. And therefore, if we find sort of those genes in patients, we can predict that those patients that have mutations in those genes probably will not respond very well to the drug. So that's how it kind of links together. And so I've done this screening now in 2 independent cell lines, and we've only just got the results recently. So I'm just going to show you very briefly that it looks like it worked. So what we found is that we found 3 kind of main classes of genes that we're interested in following up on. So in this plot, we have the most enriched guides for the most enriched genes in the 2 different cell lines, and this is showing the log fold change. So the ones in the top right corner here are the most potent resistance factors, I guess. So we found 3 genes that are involved in the STING signaling pathway itself, IRF3, TBK1, and STING itself. So this suggests that our screen works quite well. And as well as that, we've found some genes involved in intracellular trafficking, which is an important part of STING activation, as well as genes involved in epigenetic regulation, some of which are also known to be commonly mutated in acute myeloid leukemia patients. So these are the things that I'll be following up on. So the rest of the plan for this aim is to validate some of the hits from these screens by generating single cell knockouts and assessing their responses to STING agonists. We can also explore the mechanisms of resistance to try to understand better why this is occurring and if there's potential to then target this with a different drug as well. The same thing will then be done for CRISPR activation screens, and we'll take the most clinically relevant and exciting hits to validate in human cell lines to form our panel of biomarkers. So from this aim, the potential outcomes are to identify some of the very first biomarkers for STING agonist therapy, which will help inform patient selection for upcoming clinical trials and hopefully also identify targets for new combination therapies. And so overall, the goal of this project is to improve outcomes and quality of life for patients with aggressive blood cancers. I'd just like to acknowledge everyone who's contributed to this project that I've shown you today. Especially thank you to Cure Cancer Australia for funding this. I'm really excited to really get into it as well. So thank you very much.